Get started. So welcome to the May meeting for M4K Pharma. Again, we're going after brain penetrant ALK2 inhibitors for the treatment of DIPG, and we're using an open science concept to be able to do this. I put a couple of nice little pictures here at the top of the screen from our lemon face challenge here at OICR, the M4K team, and at Charles River, the team over there. So thanks everyone for doing that. I think it's a great opportunity to raise awareness about the disease, and also to raise awareness about what we're trying to do here as well. So Alex, we look forward to seeing your video as well. I'm sure you're going to have a really nice lemon face.
I think we're doing it on Thursday if we can.
Okay, perfect. Okay, so let me just move into a couple of quick slides here. So I'll just introduce the agenda. So again, I'm just going to speak for just a couple of minutes just to give you an update on the action items from the previous meeting. And then I'm going to introduce Dr. Michael Franti, who's from Boehringer. He's going to give us a nice overview of Boehringer's ALK2 inhibitor project that was targeted at the treatment of FOP. Then Jerome is joining us as well today. He's also going to tell us about some of the capabilities that he's been able to develop in the ALK2 field. And we look forward to that presentation. And then we have several short presentations from the team, from Alex, Sue, and David McLeod. And then I've got a very short presentation as well, talking about some proposals for next steps for some of the key molecules. And then Arij will tell us a little bit about how the M4K website is coming along. And for those of you who haven't seen it, I think Arij will give a nice introduction to that, and it looks quite good. And then we'll finish up with any other business. So the action items from the April 6th meeting, we had to complete the documentation requirements for the CTIP grant, and Alex, Owen, and I were able to finish that up and submit everything in a timely fashion. So thanks everyone for all of your help with that, and I'm glad to have that out of the way for at least the next 6 months. Alex was going to follow up with RBC about the assays that that perhaps SGC can transfer to RBC, and I know that that's happened, Alex, and I think that's great. Yeah. So that's fantastic. We wanted to prioritize some compounds for X-ray. David Smale, David McLeod, myself, and Alex, and I think that's been done as well. We unearthed a discrepancy between a couple of different batches of M4K 1212. We did get to the bottom of that, and the second batch of 1212 is actually the accurate structure, and it's inactive, so we don't need to follow up on that molecule any longer. I've removed it from the slides. Charles River was getting ready to ramp back up on the chemistry, and we needed to coordinate the chemistry efforts. So Sue and I have kind of communicated, and Sue will give you more information about what their chemistry efforts going to look like in the next little while. As well, we talked about accelerating some in vivo studies for some priority compounds, and so I've looked into this. We have some recommendations that we're going to propose close to the end of the meeting in terms of moving some compounds forward, and that should be an interesting discussion. We're still working on getting that MDCK, MDR1 assay online. It's close. Yeah, it's very close. And the same with our database, and compound progression tool is coming along as well. So not quite there yet with these two, but we are getting there. For those of you who haven't been part of the project, I did just want to bring back up our project flow scheme, how we're making and synthesizing and profiling compounds to move through a cascade. Basically, we have synthesis ongoing at 2 different places here at OICR and at Charles River. And the molecules get modeled and docked, and the ideas get prioritized. And then we test the analogs against RBC against ALK1, 2, and 5. And we have tested a subset of our compounds against the relevant ALK2 mutants that RBC has, and we've demonstrated that the potency is essentially equivalent to what we see for ALK2 wild type. So these are our primary screening biochemical assays. SGC is generating some X-ray structures of some of the more important compounds. And then SGC also has some cell-based assays looking at ALK2 and ALK5, and both a nanobret for the ALK2 and a luciferase assay for the ALK5. Here at OICR, we do some in vitro ADME. Certainly the liver microphone data is data that we already have, and the MDCK MDR1 is nearly ready. And then the compounds also get profiled for proliferation in DIPG patient cell lines, and hopefully looking at phospho-SMAD as well, looking at the direct consequences of ALK2 inhibition. And then, hopefully we're going to move some compounds in vivo, look at the plasma versus brain exposure. And then as we move forward, we'll move into some efficacy studies and broader profiling, but we're not quite ready to be here just yet. So that's kind of how we're profiling the compounds. As any drug discovery project goes, this is certainly subject to change and will likely change as we move forward. But this is how we've started out, and so far I think it's working pretty well. So with that, I'd like to take the opportunity to introduce Dr. Michael Franti. Michael is the Director of Regenerative Medicine at Boehringer Ingelheim in Ridgefield, Connecticut. I also had the great pleasure to work with Michael at Boehringer when I was at Boehringer in Quebec. He was the Associate Director of Virology for both the NCE and NBE projects that we had at the time. And I can tell you that he's a fantastic collaborator. He's someone that's really great to work with. We worked really well together, I think, on a rhinovirus project that resulted in some pretty interesting discoveries and an interesting publication. Michael also spent some time at Novartis and Scripps, and he did his PhD in Paris under the direction of Professor Jerome. And today he's going to tell us about the project that Boehringer was doing with respect to targeting ALK2 inhibitors for the treatment of FOP. So without further ado, Michael, I can transfer, or you can share your screen now and drive the presentation from your side.
Okay, will do. Thank you very much, Jeff. So,
thanks, Michael.
Let me know if you can see the slide.
We can see them, yeah, at least here at OICR. Yeah.
Okay, we're fine here in Pharma.
Okay. Hold on a second. Yeah. Okay. Okay, so thank you very much, Jeff, for the nice introduction, and it's always good to reconnect with all colleagues, clearly. So today, thank you very much, everybody on the line, for your time, and I will give an overview of the project that started, I would say, 2-3 years ago at Boehringer Ingelheim in Ridgefield, Connecticut, and I'm part of what we call RBB, Research Beyond Border, which is a new division created by the global head of R&D, Michel Perret. And the goal of this division is to look at regenerative medicine, but also to look at some potential other novel targets for specific indication. And this is why we picked FOP patient, thyroidysplasia ossificans progressiva, and the ALK2 inhibition. So very briefly, I will give an overview of the project, the clinical summary. I will deep dive, I would say, into the Nootropic Concept, Then what are the scientific evidences, published data that support the concept? I will also talk more specifically about all the biology we set up in the lab and also all the data we got with only published compound. Of course, as you can understand, I cannot show you any proprietary BI compound today. So I will talk about the role of IL-2 in the osteogenic and chondrogenic lineage. And I will show that how, if we can target specifically IL-2, we show that we can significantly decrease osteogenesis, chondrogenesis in vitro, and we also have some in vivo data. I will then move on to describe more specifically the target risk summary. So if we inhibit IL-2, what can happen, but also if we inhibit the other ALK, because you know there are many ALK 1 through 7, what can happen? And finally, I will end up my presentation by a short conclusion and the team recommendation that lead to a termination, unfortunately, of this project. So I don't know how you want to run this presentation, but if you want, feel free to interrupt me at any time if you have any question, or at the end of the presentation, doesn't matter.
Okay,
so
thanks, Michael.
Yeah. So in terms of project overview, so what we call the project value proposition at BI, if we can find a specific and potent IL-2 inhibitors, it will represent an innovative approach for a novel treatment strategy to reduce what we call the heterotopic bone formation, so the production of extraskeletal formation in patients with FOP. So the target class is an enzyme receptor kinase. We were using a small molecule, NCE, and we were targeting oral administration, QD-BID. And there was actually no direct competitor known in the clinic for FOP. And at that time, the key competition were Clemencia, were in phase 2 with the retinoic acid receptor gamma agonist for FOP. And there was also Regeneron, it was in phase 1 with their anti-activin A antibody for FOP. So as you can see from this picture, and I actually attended the FOP meeting 2 years ago in Boston, I mean, this disease is terrible. This patient suffer terribly. This is a rare disease, 1 in 2 million. What happened is that the child at the age of 6, 7 years old, they are normal, they are born normal, except the malformation of the big toes. And then they started to have some flare-up, so there is an important component of inflammation, and then cartilage formation and bone formation that usually started in the back. So at the end of the second decade of their life, the patient are in wheelchair, and the median lifespan is around 40 years of age. So the diagnosis is now the genetic testing with the IL-2 mutation in the 206 position. And there is no treatment, no prevention, no treatment. The supportive treatment that actually don't work are high doses on NSAID, but clearly it doesn't work. So you can clearly understand that this is a high unmet medical need. If we can target IL-2, it has a potential for breakthrough therapy. And for BI, it would have been viable only if we reach first the market. So in terms of Nootropic Concept, so as I mentioned, if we can develop a specific IL-2 inhibitors, we'll block the abnormal SMAD158 phosphorylation that I've seen actually in animal model and in patient that happen during the chondrogenesis, but also osteogenesis in the patient. And the medical value proposition is that an IL-2 inhibitors will clearly represent another approach to stop and reduce the heterotopic bone formation in patients affected by this terrible disease. So what are the scientific evidences that support the new therapeutic concept? So there are many data in the literature. So here I'm going to talk about FOP, but also heterotopic ossification, the non-genetic form, because it's basically the same physiopathology. So of course there was the SEVR1 genetic mutation discovered by Ellen Shore in Fred Kaplan's lab at UPenn. And they show that the 206 mutation in ALK2 clearly trigger the FOP disease, which is an extreme form of heterotopic ossification. In addition, and this was a poster by the Walter Reed National Military Medical Center, they show that patient soldiers that underwent reoperation for HO excision after war wounded, there was an upregulation of ACVR1 among 5 other genes, but clearly ACVR1 was upregulated. Data in animal model, in vivo and in vitro, also from the literature, are consistent with the link to ALK2. So with this LDN compound 193 that actually we used, and you're going to see that later in my presentation, they show that an inhibition of ALK clearly mediated the SMAD158 phosphorylation and decreased HO in the mouse model. In addition, these two compounds, the LDN193 and LDN212 that we use as well, in an FOP mouse model, so this FOP mouse model is a knock-in with 206 mutation and clearly mimic what you can see in the patient. You could see that if you inhibit ALK2, you clearly inhibit the SMAD158 phosphorylation and you decrease extraskeletal formation. And finally, in the mouse cells, so they use a mouse embryonic fibroblast cells, they show that if you can inhibit ALK2, you can clearly inhibit the chondrocyte differentiation from the MSC into chondrocyte. Finally, there are some published data in human cells, and this, I believe, is a backup slide. But Levy and collaborators, so we collaborate with Levy, Dr. Levy, and I will show you the data. He showed that he was able to take some mesenchymal stem cells from burn patients, and he showed that there was an upregulated of the phosphosmad158, and that trigger a more drastic differentiation into osteogenic lineage. And finally, I'm going to show you some in-house data. So what we did, we didn't have any knowledge in mesenchymal stem cells. So first of all, in the lab, we set up the human bone marrow-derived mesenchymal stem cells. And the first question was, can we differentiate them into osteogenic lineage? And can we look at the ALK2 family gene expression? So what we did, we cultivate the cells in growth media for 14 days. And then for 12 days, we switch into incomplete differentiation media, but supplemented with BMP6. We add BMP6 at day 4 and day 8. And then we measure by qPCR the level of expression of ALK1 to ALK6. And you can see that the more the mesenchymal stem cells differentiate into osteoblast, the more you see an increase of expression of ALK2, but also ALK5. So clearly we can say that if we target ALK2 on the mesenchymal stem cells, clearly the target is here. Then we move on and try to select some siRNA, specific siRNA, to inhibit specifically the ALK2 gene. I'm not going to show you the data, but we tested 22 different siRNA. We found one that was specific against the ALK2 gene. And we did the same experiment. We used mesenchymal stem cells, human mesenchymal stem cells. We did one transfection of siRNA. Then we follow the same protocol than before. We add BMP6 at day 4, day 8. And at day 12, we measure the expression level of ALK2. And we measure also two biomarkers, alkaline phosphatase and osteric gene, that are actually a biomarker for osteogenic differentiation. And here you can see the ALK2 gene expression with different concentration of BMP6. So as a control, we use an irrelevant siRNA. We don't see any decrease of ALK2 expression as compared as if we use a specific ALK2 siRNA. So clearly the answer here is that the siRNA was working, can clearly decrease the level of expression of ALK2. Now, does this translate into a lower differentiation of osteoblasts, of osteogenic lineage? The answer was yes. When you look at the IL-2 gene expression or osteogenic gene expression, which are biomarkers for the osteogenic differentiation, you can clearly see a significant decrease of the gene expression alkaline phosphatase and osteorex in both cases when we use a specific IL-2 siRNA. So clearly the answer from this slide is if you can inhibit the expression of IL-2 at the surface of the mesenchymal stem cells, you can clearly inhibit the differentiation into osteogenic osteoblast. Then we move on and we perform exactly the same work, but this time looking at chondrogenic differentiation. So once again, we use the same protocol here. We use BMP6. We looked at the IL-2 gene expression with the scrambled siRNA as compared to the IL-2 siRNA. Clearly, you had a very nice decrease of the expression. And this time, we looked at the COL2 gene expression and alkaline phosphatase gene expression, which are again biomarkers, but this time to follow the chondrogenic differentiation. And you can see the effect was less clear as compared to the osteoblast, but clearly you can see a decrease of the role of the chondrogenic differentiation when you inhibit IL-2, and also using the alkaline phosphatase gene expression. So here, when you inhibit IL-2 with an siRNA, inhibit the expression of IL-2 with an siRNA, you can clearly see that we decrease the chondrogenic differentiation. Then, so the conclusion from these two slides that an IL-2 expression is essential at the surface of the bone marrow derived mesenchymal stem cells to differentiate the mesenchymal stem cells into osteoblast and chondroblast. So then we use a published compound, the LDN compound, so the LDN 193 and 214. So we also set up some IL-2 enzymatic assay. You can see that they have similar potency, nanomolar. We set up, of course, the phosphosmad-1 and phosphosmad-2,3, which is not represented here. And we set up also the ALK1, 3, 4, 5, and 6 enzymatic binding. And you can see that the LDN 193 seems to be more potent as compared to the LDN 214. And we have also all the different parameters that we use actually for in vivo study. You will see that later. So what we did, we started with the LDN 214, and we wanted to see if this LDN 214 was able to significantly inhibit the differentiation of MSC into osteoblast. Once again here, we looked at the alkaline phosphatase gene expression, and here you have the concentration of the compound. And again, we looked at the osteorex gene expression. You can clearly see a very nice dose response of the compound, meaning that this compound, even if it was not a specific anti-IL-2 inhibitors, but this compound was able to strongly inhibit the differentiation of osteoblast from the MSC. And this is the EC50, the IC50 here. Then we move on and perform the similar experiment, but this time using an adipocyte differentiation, because I'm sure you know that mesenchymal stem cells can provide chondrocyte, osteoblast, skeletal muscle also in some case, but also adipocyte differentiation. And this actually was never shown in the literature to my knowledge. And we use exactly the same compound, the LDN214, and you could see that we were able, by following PPAR gamma, which is a biomarker for adipocyte differentiation, we obtained a very nice titration curve. And this is actually a picture of the droplet. You could see here the droplet, the fat droplet, and pretty much no fat deposition here. And the EC50 was actually similar to what we obtained during the osteogenic differentiation. So we were kind of surprised by these data. And this immediately triggered a question that I don't know if you had the same, but if you inhibit an IL-2, you might actually inhibit the adipocyte differentiation, and therefore your fat in the body should go somewhere. And I was talking to our NDS colleague, and they said that this could be a disadvantage, something that could be quite detrimental, because the fat might accumulate in the liver. And this is something we can talk at the end of my presentation if you want.
Okay.
Then we finally look at the effect of this compound 214 into chondrocyte. And here we're looking at the chondrocyte collagen deposition. And you could see here a beginning of a dose response of 1.1.1 with the LDN 214. So clearly this tool compound that is published was able to target IL-2, not specifically, but has a strong binding to IL-2, and was able to inhibit the osteoblast, chondrocyte, but also adipogenic differentiation. Something also that we were interested is to compare the tool compound to the compound from the competition. So first of all, we tested against NSAID inhibitors ibuprofen. Why? Because we heard, our medical colleague heard that FOP patients were treated with massive dose of NSAID, and it was working, which clearly it's not working in the clinic, but I don't know where do they come from with this idea. So we tested the NSAID inhibitors in red here on the osteoblast differentiation, and you can see a flat line. Of course, NSAID inhibitors do not inhibit the differentiation of MSC into osteoblast. We also tested palovarotene, highly selective retinoic acid gamma, a receptor gamma. And palovarotene was in brown here, and there was no inhibition of palovarotene to the differentiation into osteoblast. We tested also in green the anti-SMAD2/3, and there was, of course, no inhibition. We know that the differentiation is triggering via the SMAD158. Something also not represented here, and I couldn't find the curve. There was one investigator in Japan saying that rapamycin mTORC1 inhibitors, mainly mTORC1, was able to inhibit the differentiation of MSC into osteoblast, and actually he tested in 2 FOP patients, Japanese patients. And we show that rapamycin does not inhibit the differentiation of MSC into osteoblast. And actually, there was a press release from his company or his academic lab, I don't remember. There was a press release saying that actually the 2 FOP patients treated with rapamycin they don't show any improvement of extraskeletal formation, so it doesn't work. Then we move on and perform the same test, but this time looking at chondrogenesis. And so this is not osteroids here, there's something wrong here. Yeah, this is COL2. And surprisingly here, the brown curve here, this is palovarotene. You can see that palovarotene was extremely potent, much more potent than 2 compounds that can inhibit IL-2, as inhibiting the chondrogenesis. So this is something that started to, I would say, worry us, BI, because as you may know that when extraskeletal formation starts, it starts with cartilage, then after became bones. So if palovarotene was able to strongly inhibit cartilage formation, that could have a positive impact, I would say, for the patient and in the clinic. And this is something I'll talk later. And then we moved on and established a collaboration with Dr. Levy, as I mentioned before, at University of Michigan. And Dr. Levy developed a very elegant model of heterotopic ossification mouse model. It's not an FOP. So what he's doing is basically burn 30% total body of the back of the mice, and then he injured the Achilles tendon. And he used these 2 compounds, the 214-193, IP 6 mg per kg, and the bone were measured 9 weeks after the injury. So those are the data of the bone density, and you can see the PBS control And you can clearly see the LDN 193 and 214 strongly decrease the bone density, the extraskeletal bone formation. And as you can see on this picture here, worth 1,000 words usually, the PBS, you can clearly see the extraskeletal formation and a significant decrease of the extraskeletal formation with the LDN 214 and 193 as well. In addition, what he did we didn't ask him, but he did it, and I think it was an interesting data. He performed a similar experiment, but he used the compound this time 2 weeks after injury. In the previous slide, the compound was injected at the time of injury. Here, the compound is injected 2 weeks after injury, which mimic, I would say, more what can happen in the clinic. And you can clearly see that 2 weeks after injury, the LDN 214, even 4 to 5 weeks, was still able to strongly inhibit the extraskeletal bone formation. While for the compound 193, there was still an inhibition at 2 to 4 weeks, but no inhibition at 4 to 6 weeks. And of course, he looked at the SMAD 158, and there was a strong inhibition of the SMAD 158 using the compound LDN 214. So the summary of the biology that we run in my lab, was that clearly if you can target IL-2 inhibition, so with a tool compound, in human MSC, you can clearly inhibit the osteogenesis, chondrogenesis, but also adipogenesis. So for adipogenesis, is it a problem? This is something that we were, I would say, actively discussing. We showed also that NSAID, pyrrolidine, does not inhibit the MSC differentiation into osteoblast. However, as I show you, pyrrolidine was extremely potent to inhibit the cartilage formation from MSC. We also show that the LDN 214 and 913 clearly inhibit a strong significant effect, clearly exhibit a strong significant effect on reducing the formation of bone into this burn tenotomy mouse, and this was a collaboration with Dr. Levy, University of Michigan. And the next step would have been, because the project has been terminated, the next step would have been to use an antivirus to bring the 206 mutation into the human MSC, and we actually pretty much got the MSC. Why did we use an antivirus and not CRISPR? Because with CRISPR for MSC, it's pretty much impossible to do because you cannot start from single cell with MSC. So we had to use an antivirus. And of course, we initiated a collaboration with Kaplan, Allen and Shore at University of Pennsylvania. They developed, I would say 6 months ago, an FOP genetic HO animal model. So we're ready to validate the model with them in order to initiate the same in vivo study with the tool compound. In addition, we also perform some target risk summary. So we know that ALK2 is required for the implantation of the blastocyst. So it's critical during embryogenesis. So this is clearly that's something that we would have excluded from a clinical trial. So the woman that can be pregnant. But again, if you think about FOP patient, I don't think FOP patient woman, unfortunately, can be pregnant. And this is also something we were carefully monitoring because IL-2 is required for iron homeostasis. So the inhibition of IL-2 may lead to iron overload. So clearly, a selectivity for IL-2 was highly desired. But I would say at this stage that it was a mistake because we could have been clearly with an ALK1 and IL-2 inhibition. Why? Because ALK1, the safety margin was possible. ALK1, there was some vascular effect if you inhibit ALK1 due to pro-angiogenic effect. There was some development reproductive toxicity, but clearly the safety was acceptable, I would say. However, we should absolutely not, we didn't want to hit, of course, ALK4 and especially ALK5. Why ALK4? Because there's a muscle atrophy, based on myostatin signaling through IL-4. So clearly FOP patient, if you decrease their muscle mass, that will not be good. And IL-5 should absolutely not targeted because of cardiotoxicity. In terms of competitive overview, at that time, there was a lot of preclinical competitive compound, so targeting IL-2, but also there was one company in Japan with an IL-2 specific monoclonal antibody. We produced this antibody, and indeed, it was extremely specific, extremely potent. And of course, there was the related competition, so the Regeneron antibody, the anti-activin A specific that actually trigger SMAD158, but only on the IL-2 mutated IL-2 206, which to me is still surprising. You had also this soluble SCVR2, so soluble IL-2, the ligand trap, what we call, and the SCVR2 antibody from Novartis. And of course, there was this unrelated competition from Clementia palovarotene. And what happened this year, beginning of this year, Clementia published some great result for the patient, showing that when they use their retinic acid receptor gamma agonist, they show a 75% reduction in bone volume formation in patients. And it was clearly a favorable clinical safety profile that support a chronic use. And they were ready to move, and they actually started to move into 80 patient phase 3. So with this data, BI decided to stop the project. And what I can tell you today is that there's a clear link for FOP patients between IL-2 expression and the bone formation. As I mentioned, I cannot show you, and it's frustrating sometimes, I cannot show you the chemical matter, but we were able to reach an early status, what we call the mid-pre-lead optimization. Our compound was not specific against IL-2, it targeted IL-1 as well. And at the beginning, we were really focusing on a specific IL-2 compound, but I can tell you that today, if you would start it again, we would focus on IL-1, IL-2, because IL-1 is safe, especially for FOP patients. And as I mentioned, the competitive situation was challenging. Indeed, palovarotene published some very exciting data for the patient. So this is why the IL-2 FOP patient was terminated 7 months ago. I think this is the end of my presentation. Thank you very much for your attention, and I will be happy to take any questions.
Well, thanks, Michael. That's a great story. You certainly generated a lot of data, and I think from our perspective, LDN214 is the lead molecule for our effort here. So it's very encouraging to see, how much you were able to do with that compound and how much you were able to validate the ALK2 biology with it. In our hands, it's interesting because we find that compound is a little bit less potent than what you've highlighted there. I wonder if you can go back to the slide where you talked about those two compounds.
Yes. Are you talking about in vivo or in vitro?
In vitro. Yeah, I mean, I have some questions about in vivo as well, but the in vitro potency, where you have the two compounds side by side. Yeah, or
3 and then 5.
Yeah, I think even just up even further, where you had just the intrinsic potency against ALK2. So yeah, it was
5.3. 5.3.
Yeah.
And
There you go.
The previous slide was 3.
Yeah. So
This one?
yeah, yeah. Yeah, our potency that we measure is a little bit less than that. Yeah, we're getting in our ALK2 assay about 100 nanomolar for 214117, and then in the cell-based assays we're getting about 200 nanomolar, which I think lines up with.
Yeah. So it's a SMAD1. It's a reporter cell line you used.
Alex, I don't know. We haven't, I mean, phosphosmad-1 has been looked at with this compound previously in the literature. And I don't know, Alex, do you want to talk about your nanobret assay and how, because we have an ALK2 inhibitor, Alex Bullock at SGC, has an ALK2 assay that is based on nanobret technology that measures the cell-based potency.
Off the top of my head, I don't remember the numbers. I know the published ALK2 in vitro kinase IC50 was around 25 nanomolar. I'm not sure what reaction biology data, what the IC50 we have from them for this. I think it was lower still, like, was it 80 nanomolar?
It's about 115. Yeah, 100 nanomolar. Yeah.
And then the cellular assays, probably the PSMAD1 reporter is very similar to the BMP response element reporter. Maybe it's the same thing.
Yeah.
The dual luciferase. Yeah.
Yeah, we were using the same. We developed this assay in house here, the luciferase based reporter. Yes. Yeah. Yeah.
Cellularly consistent.
Yeah, cellularly, the cell-based data is, is pretty consistent. I mean, we're at about 200 nanomolar, and that's 140. So that's we're hoping in our project to get compounds more potent than that, but I mean, it looks encouraging. The HERG for that compound is 3.5 micromolar. Okay, and then did you guys run any in vivo pharmacokinetic experiments to correlate the in vivo results that you saw?
Yes, we did. I didn't add it here. I don't remember. If you want, I can send you the slide later on. I don't remember at the top of my head, but again, this is published compound. Yes, we did. Yes.
Yeah, that'd be great if you could do that. I think it's always valuable information, right?
Yeah. We'll hold on a second. Let me take note. Yeah, we'll do that. Yeah, I know we did. I have a small presentation somewhere. Yes, I know. Yeah.
Okay, fantastic. Yeah.
A quick question. With regard to your differentiation phenotype in osteoblast, does ALK2 affect osteoclast differentiation in the, because you rely on homeostasis between those two processes for bone formation. So does ALK2 affect osteoclast differentiation over?
What do you mean? So if you inhibit ALK2, you inhibit the osteogenesis from osteogenesis. I didn't hear your question very well. Sorry about that.
Yeah, so ALK2 looks like it inhibits, the osteoblast differentiation.
Yes, correct.
I'm wondering if it also now increases osteoclast.
Oh, osteoclast, yes. So that's actually something we started to look at. And as you can imagine, since you're asking the question, I'm pretty sure you know what you're talking about. It's a very difficult assay to set up osteoblast, osteoclast. And this is actually something we are ready to set up. We don't have any data. The assay was up and running, and then we canceled the project. I would expect, yes, to perhaps produce a disequilibrium between osteoblast and osteoclast. I would say yes, perhaps, but that's a very interesting question. Yes. Yeah.
Okay. Thank you.
Yeah, I also thought it was, very interesting and informative, certainly for our project, the fact that, because, I mean, we've been profiling ALK1, and certainly within the 214, 117 series, we've been having a very difficult time trying to generate any kind of specificity against that kind of.
I don't, personally, I mean, I think if you target ALK1, ALK2, it will be fine. And again, we're talking about DIPG. This is a very extremely severe disease. So personally, I do think if you have ALK2, ALK1, it will be fine. ALK5, no, of course. What do you make of this story, inhibition of adipogenesis? Because when I mentioned that to my colleague in non-drug safety, the department, they told me, well, you really have to be careful. If you inhibit adipogenesis, the fat produced by the body will have to go somewhere. and most likely will accumulate in the liver. So are you concerned about that in your indication?
Well, I mean, I think it's something, you know, to be honest, it's the first time I've thought about it. I think that it's something that we should think about. I think in you know, certainly in childhood cancer and DIPG, the patients really don't have any other options.
Yeah, of course.
So, you know, I think that, you know, certainly in that format, we could get away with a little bit more, but I guess it depends on how serious that issue is and where it ends up being deposited. It's an on-target potential. It's ALK2 that is in the adipocyte differentiation pathway that, you know, could lead to fatty liver.
Did you see anything in your multi-dose animal model? Like, did you do any multi-dose animal model, and did you see anything on that regard?
No, I asked Dr. Levy about that. He didn't see any fat decrease in the tissue, or he didn't see any effect on the liver, and neither on the muscle formation, because I was also a little bit worried about that. But no. But again, no, this is 9 weeks.
Okay.
So up to 9 weeks, it was okay.
Yeah.
Thank you.
It's interesting.
It is another thing that affects some other mechanism.
It's possible, yeah. Okay, does anyone else have any other questions for Michael? Anyone on the phone? Okay, well that was fantastic, Michael. You know, thanks so much. I mean, it's really great that you were able to present that, and that Boehringer was able to allow you to present that. And you know, certainly on behalf of M4K and OICR, we thank you greatly. And certainly, you know, we hope you'll remain as part of the team and be able to attend any meetings that you're able to attend, and contribute to the team as much as you can as we move forward.
Yeah. Thank you very much, Jeff. And I would love to. I mean, I'm going to travel quite a lot, but as much as I can, I will dial in and participate to your teleconference and progress of your research to find a cure for this, terrible disease again. Thank you, Jeff.
Thank you again, and we'll talk soon. Okay.
Yes.
Okay. Fantastic. So I guess next on the agenda is Jerome. So Jerome is originally from Montreal.
Yeah.
Yeah. And he spent a lot of time at McGill, and he's in Toronto more recently at UHN. And he also has a very keen interest in ALK2 and the ALK2 biology, and he's, I think, put together some pretty interesting assays in an animal model that potentially we can tap into. We put together an MTA already, so he's willing to test compounds for us, and we just have to come up with a list, and hopefully by the end of today's meeting we can do that. So with that, I'll pass you the mouse.
All right. Thank you so much. Nice to be here. I just want to give you a slight overview of what I've done so far. This was basically aimed at understanding how did ACVR1 mutation drive DIPG. So to do that, I engineered a conditional knock-in mouse model where the most common DIPG causing ACVR1 mutation, the G328V, can be conditionally activated in any cell type with the action of recombinate. So just to put it in context with FOP that we just heard about, this particular mutation, that's the most common one in DIPG. It's actually never been reported in FOP. There's some very few patients that have glycine 328 mutated, but interestingly, apparently never to a valine. So there might be some interesting, biology there as well in terms of differences. So this is the mouse model, and yeah.
Excuse me, we've not got the slides at the moment.
Oh, okay.
Oh, hold on. Let me see here. Hold on.
Sorry to interrupt.
Oh no, I'm glad you did. Let me just go back. possible I didn't share. All right, share screen. And share my desktop. Can you see them now?
No, we've still got the sorry, no, we've still got the screen up. That
Which one is it?
You click on share.
is it just me, or has anyone else got? Ah, that's coming. Yeah, that's better. Thank you.
Okay, yeah, let me just make sure that, there you go. You can see this one now? A novel conditional blockage? Okay.
Yep, that's great. Thank you.
Perfect, okay. Sorry about that. All right.
So yeah, so to validate this mouse model, basically I first activated the mutation in the whole body using a deleter Cree mouse. That's the mouse where Cree is active in the whole body, and this is lethal either in embryo or around birth. Just recovered a few stillborn animals with severe developmental anomalies. But of course, the main interest was to look at what's happening in the brain. So I targeted the mutation to oligodendroglial progenitor cells. Those are possible cells of origin for DIPG, so using olig-2 Cree. And this is just a summary of the salient findings that we got so far. This is part of a manuscript that hopefully I can submit soon. There's a certain level, a small level of early postnatal lethality, but mice develop neurological anomalies when they're in young age. This is associated with oligodendroglial cell hyperproliferation, as well as very interestingly, lineage differentiation arrest. So that's conceptually different than what we just heard about in FOP. But this process is thought to be commonly a key initiating event in gliomagenesis. So in that context, it's interesting. That mutation by itself, at least in the olig-2 Cree, does not seem to be sufficient to induce brainstem tumors, even if I add a histone K27M mutation with it. But currently, I'm adding additional drivers that are relevant to ACVR1 mutant tumors, so PIK3CA and PTEN. These are early days. I recently had a quadruple mutant mouse, a young mouse with developed a brain tumor that we're characterizing right now, and there are more animals coming. So I'll know more about that in a few months.
So even with the K27M, ALK2 mutant wasn't enough to initiate.
That's correct, yeah. So maybe, again, this is one model with olig-2 Cree.
Right.
Could have to use different Cree drivers. This is something that we're looking at. Could need more time, more mutation, all of these things. Yeah, that's possible. I mean, there's currently no mouse models of ACVR1 driven DIPG tumors, so we're still kind of trying to figure out what's the right combination of mutations in mice, at least.
Right.
So just to show you kind of in general the type of thing that we could look at with your compounds, this is just some work that we've done in collaboration with Peter Ten Dijks in the Netherlands, and Alex Bullock is involved in this as well. To look at the effect of a compound called E6201 that was interesting for us because it targets ACVR1, but also maybe PDGFR signaling, which is thought to be a key signaling mediator of tumorigenesis in DIPG. This is a covalent inhibitor. So a type of thing that we can do, for example, we can use our mice and look in primary cell culture, so prepare glial cell culture from early postnatal brainstems, and activate the mutation by transducing the cells with an adenovirus that express Cree, and then we can add the drug and look basically if we can block the signaling induced by mutant ACVR1. And this is just giving you an example with one well-known DMP target gene, ID1, where you can see that turning on the mutation and the conditional mutation stimulates expression of ID1. If you put the ligand antagonist login, that does not block it, presumably because it's BMP ligand independent, but E6201 largely blocks it, and with or without noggin. So what we can do with that as well is with this type of compounds, we can test it in patient-derived cell lines. So these are 3 cell lines that I got from Stanford University. They have a nice program there to derive patient cell lines for DIPG. So here I have 3 cell lines. 2 of them are CBR1 mutant, 1 of them is not. And you can look, you can see, for example, in the case of EC201, that the dose dependency decreases the growth or the viability of these ACVR1 mutant cell lines. So this is kind of like a simple assay that we can use in a medium to high throughput manner and test some of the compounds that you are developing.
How long is the proliferation assay? Like
this is a 4-day assay after 4 days after drug addition. Yeah.
Nice.
And the last thing that I want to touch on is that I'm currently optimizing a procedure to transplant these cells and xenograft. So this is NSG mice, so we can inject in the postnatal day 2 brainstem. This has been reported by people that derive these cell lines, that at least many of these cell lines can successfully implant and give brainstem tumors in this context. So this is early. I have 2 cohorts of mice that were injected with these cell lines, and I'm following them right now. So hopefully I have this as well as a tool to eventually test in vivo. Yeah, so that's basically what I have to say for now.
Yeah. What was it company added this slide before for the growth profile?
It was the E compound, the dual PDGFR alpha.
It's a MEK1. It was developed as a MEK1 inhibitor, but it's a bit of a dirty inhibitor, which may be good actually in this context.
So you have never tested any like ALK2 inhibitor in this assay?
Yeah, so I haven't done it personally. That was the work of Peter in the Netherlands, looking at ALK2 inhibition directly. For me, I just used it and basically in the context of the primary glial cell culture and then the patient-derived cell.
Okay.
So it's not clear what the mechanism is of the inhibition?
No, it's not actually. But I know Alex is currently trying to, and I think has been successful in crystallizing it with the receptor.
Yeah.
So we'll know.
Yeah. We have some data, certainly for the lead compound 117 on the DIPG4 cell line. That's the one where the efficacy study was done, I believe.
So anytime he's testing it?
What's that?
He's testing.
Alex, I believe, has some of these assays as well, so it'll be good to kind of have it happening in a couple of different labs. So I think Alex will speak a little bit more to that when in his presentation, which is actually next. So let me just pull up.
It would be good to combine in a PDGF and an ALK2 in the test.
Yeah. Yeah. Oxford.
Thanks, Jeff. Thanks, Jerome. Very interesting data.
Yeah.
Jeff, can you change the slides for me when I shout?
I sure can. Yeah. Yeah. You. Everyone can see this. Yeah.
Yeah, no problem.
Thanks. Okay. So as Jerome was saying, from the last M4K meeting, we were growing crystals for maybe half a dozen compounds that Jeff had identified and the team had identified as high priority ones for structures. We collected data just a couple of days ago at the synchrotron. So fingers crossed, we'll have 2 new structures in the next week or so. So the compound on the left, I think, is a very interesting compound. Even in cells in the nanobret assay, this is ALK2 IC50 in HEK cells, it's 30 nanomolar. So that's a really nice compound. Look forward to the structure of that one. And then Jerome had mentioned this E6201 compound, this macrocycle. We also collected a 1.6 angstrom data set on that, so we'll be interested to see how that binds. And then we've got, sorry?
Do we know the potency of 6201 against ALK2?
I've not tested it. I think Peter in Holland is doing that. So
I think
we've got.
Yeah, I think it was consistent with the IC50 that we saw in patient cell lines, like something around, I don't remember, like 0.4 micromolar or something like that.
Okay.
Yeah.
Yeah.
If I remember correctly, something like that.
We've got some more crystals ready to go to the synchrotron trip. Do you know which day it is, Ellie? Tuesday? Next week?
14th.
Well, in a couple of days, anyway. So we're hopeful that we can get a couple more structures. So hopefully we present these structures at the next meeting. Next slide.
Great.
So I've just put together a couple of slides summarizing the nanobret assay. So in green here, these are all out-2 assays in hex cells. And Jeff and the team were asking us just to check reproducibility of the assay with independent replicates. So each curve, I think, is a set of technical triplicate experiments, and then the different lines are independent triplicates. So I think overall here, there's quite good agreement. And again, all these compounds fairly potent. Next slide. I think you'd identified one compound where we had a discrepancy where the nanobret IC50 was much different to the in vitro kinase reaction biology IC50. So we've repeated these in 3 experiments. And again, I think there's reasonable reproducibility there too. I can't remember what the reaction biology IC50 was. Anyway, next slide. Okay. We also tested compounds that were made in different synthesis batches. And of course, fingers crossed, as you'd expect, you get good agreement there too. Next slide. On this slide, I just highlighted a couple of compounds of interest. Apologies for the drawing on the bottom left. That's just pulled from our database, and it obviously has a headache that day. I think we can call this a kind of benchmark compound. So this is with the amine on the pyridine, so the aminopyridine. So that's our most potent compound in this series, with sort of in cell activity, IC50 is around 5 nanomolar, which I think is quite a good control compound to test against. And then I was also interested in the compound on the right, where we've lost the trimethoxy completely. And there we've got slight scattering, but IC50 is in the range of 100 to 200 nanomolar. So again, I think I'll come back to that, but that's quite an interesting compound. Next slide. Moving on to some of the new compounds that OICR just sent. These were the 2 most potent compounds, I think, John. Yep. So one where we have the fluorine in the middle of the ring there, 100 nanomolar, and then with the metal on the aminopyridine, also good potency there. Do you have any explanation for the error bars there, Jong-Soo? No,
it could be a fluke, like with the machine, but
okay.
Yeah, I'm going to.
You're repeating that, okay. So we'll repeat that to see if we can get rid of those error bar problems.
Yeah.
Next slide.
Sorry, just a comment here on 205A. We were a little bit surprised that inside methyl group makes such a big difference in terms of the nanobret value. We weren't expecting such a drop off, so hopefully that it's the machine variability that can explain that, right?
Right, because when when the NH2 is a hydrogen, it's essentially equal potent, right? Whether or not you've got the methyl there, or not.
So in this compound, we've gone. If we take away the methyl, we go to 5 nanomolar in the nanobret. So we've basically got 10-fold drop off.
Right.
So Alex, do you know
what
methyl is doing?
What the methyl is doing?
Yes.
Well, hopefully our crystal structure in a week or so will give some insights, although that's with a different solvent group, solvent exposed area. But I think the hypothesis was whether it impacts the rotation of the ring, the trimethoxyphenol.
Have you
I mean,
tried
205, 205A may be a unique example, a unique situation, because now we have two ortho positions basically to that trimethoxyphenyl, which are hindering its rotation, right? So that may legitimately be a log drop off in the cell base, but we don't really know yet until the data gets a little bit tightened up, right?
Right. We don't have the intrinsic potency yet for
Right.
RBC for that yet either, right? So that'll, I think, also shed some light on that. So, yeah. Yeah.
Have you ever tried any electron withdrawing groups in that position?
Electron withdrawing groups at this position?
Yes.
Yeah, that's in the plans. Yeah. We're also looking at electron donating to try to improve the hinge binding, but
yeah, because we have a similar, well, in a different series, we have changed the nitrogen in sort of a similar position, and we lost affinity, and we think it's because we sort of have too much electron density in the ring.
sorry, can you repeat that? We kind of
We have a different series where we removed one of the nitrogens, and we lost the affinity. And we actually think it's more electronic than the rotation in that case. So we're thinking about putting an electron withdrawing group in a sort of similar position. I mean, the structure is different, but I think it's similar to what we are.
Okay, yeah, it's interesting, and I think we'll come back as well because you know, we certainly and we talked about this a little bit before, Alexi, about sending some of our compounds for your RIPK2 assay, right? So we can, I think we're ready to send some of the analogs that we're interested in having you test, along with the ones that you're interested in as well for the RIPK2 story.
Okay, then these are just less potent, but a few more selected examples from the latest shipment of compounds. You had an attempt on the left to replace, modify the hinge group, so that unfortunately hasn't been successful, although we don't have the perfect trimethoxyphenyl on that one. And then some of these sulfonamide type groups, Alexi has had success with, and we put those through the nanobret as well. The middle one is the most potent compound. Out of this series, I guess there's going to be some trial and error, sort of probably can't take too much difference between 375 and 286. So these were not as potent as I was expecting. So maybe, Alexi, you could have a look at your data and see if these make sense to you.
Those are without the 2-amino stuff as well, right?
This is
yeah.
right? This is in the cell.
This is
in the cell.
Yeah.
Yeah. I mean, I think in vitro there was definitely more potent.
Yeah, I think we'll see. You know, they've been submitted. We're just waiting on the data, and it should be in any day. And we're actually surprised it didn't arrive yesterday. That's often the case.
One thing that you have, are these the compounds from Greg, or those are the compounds that you synthesize? Because the compounds from Greg should have an amino group.
Right. Right. The compounds from Greg would have the 2-amino, but in our hands, that leads to much lower selectivity against ALK5. So we're trying to stay away from that.
So we haven't tried any compounds without either methyl or amino in that position. So in terms of IC50s in vitro that we got from reaction biology, I would expect it certainly to be higher than that.
Okay. I think we'll wait for the reaction biology data and see how they compare.
Yeah. But it's
all the photon in the middle position, he gave us around, well, anywhere from 5 to 50 nanomolar. Yeah.
Right. Yeah. And that would be consistent with the drop-off in potency that we see as well by removing the amino.
I think there'll be a drop-off from removing the,
and that,
sorry.
And then 4-methoxy.
Yeah, removing that group might help, might change.
You're more potent without having the H there. So
But keep in mind, all of these compounds with the proton in the middle position, they all have a significant drop-off in cellular activity against RIP2 as well. So that's what we saw for RIP2. We saw IC50 against recombinant kinase being a lot better than nanobreath.
Okay. Next slide. This is just comparing two different types of cell assays for ALK2. So on the left column is the nanobreath assay, and then the DLA refers to a SMAD1 kind of binding element, the VRE luciferase reporter, so transcriptional reporter for downstream SMAD target gene ID1. And in general, there's quite good agreement between the two assays. So I think the nanobreath as a target engagement assay is quite reliable. It's a lot cheaper, a lot faster, a lot more easier to run in a kind of higher throughput mode. So I think we prefer that if we can get it as our benchmark first round assay. We've been talking with ProMega quite a lot about extending the nanobreath from just ALK2 to ALK1 to 6 as a complete panel. That's a work in progress. They're helping us with potential tracers to help that. I've just flagged on the right, or Jungfu has flagged for me, one example on the top of a compound where we had a disagreement between these two assays. And maybe that's the quality of the signal in the transcriptional reporter on the right. So maybe we would need to look into that. I know we were having trouble with activin and whether our batch of protein for inducing signaling has gone off in some way. Because normally we can get signals up to 30 on the y-axis, and there we're at 7 or so. So that's the only outlier. Next slide. We also obviously had some failures, so some compounds that didn't seem to give binding. I don't know if you have any particular comments for us to repeat any of these, but do let us know if you do.
I think the intent here, Alex, was that we were looking for something which was very structurally similar to our lead or the parent in the series, and we could use it as a negative control, right?
That would be good for us, yes.
Right, so I think that 2040 would be an ideal candidate because it's so structurally similar and just moving that next one position, right?
Yeah, I like that. That's a good idea.
Yeah.
Okay, next slide. Chongfu's also been generating data from time to time with ALK5 when he gets the time. Just highlighted a few selected compounds here. The compound on the far right with the funny drawn structure, that was the 5 nanomolar compound in the ALK2 assay. And as you can see with the 2 amino, it's not very selective, so it's 40 nanomolar in the ALK5 assay. In the second from the left is the one with the methyl. So that shows reasonable selectivity. And then the one I boxed in blue, I thought was quite interesting. And I'll come back to that in a moment, because obviously there we've got 10 micromolar ALK5 activity, and I think we were at 100 to 200 nanomolar ALK2 activity. And then we've been exploring tracers on the bottom for ALK5, but the ALK2 tracer doesn't work. So then, next slide. So this is just my one summary slide, the final slide. We haven't done much with this compound in terms of exploring that, but that might be one you might want to consider.
Yeah, no, I noticed that as well, that the potency of that compound in the nanobreath assay is quite good. It's interesting because in the RBC against ALK2, it's 180 nanomolar, and ALK1 it's about 130 nanomolar. So, it's interesting because there's really kind of a one-to-one relationship between the intrinsic potency and the cellular potency. I think, it'd be interesting maybe to try to get an X-ray structure of that compound. It could bind either way, yeah.
Okay. Okay. Yeah, we can do some more orthogonal assays on that one.
Yeah.
So the second piperazine, where do you think it's sort of pleasing?
I think that would just run out towards the front of the ATP pocket.
Well, that's
The question mark.
Yeah, we're not 100% sure about that yet. I think that's why getting an X-ray structure, would help. Maybe introducing a methoxy,
would it
as
be
well.
interactive with
Kind of position there.
Like that.
Right, and they were worse.
Awful.
Yeah.
You can also check the level of nitrogen in the middle wing by one position. Most likely it's the important one.
Yeah, this one,
Most
that's
likely.
the hinge binder, yeah.
So should we hold off and get a crystal structure before thinking about chemistry?
Yeah, I think, that would definitely help us, because right now we're thinking it could be binding the other way. I think there could be some pretty significant changes, so it would be interesting to see how that's binding to try to drive the chemistry forward.
But Doug, did you have any other analogs with the pyridine in the 4th position?
The pyridine here?
No, in the 4th position, the 4th position. So the same compound, but without the piperazine.
Yeah, I made one with the morpholine as well, and it was totally dead, as far as I can remember.
For example,
and
one in that position.
Yeah, and a few others with methoxypyridine and various assorted analogs.
They were not active.
Maybe 5 to 10.
Yeah.
This was the only one that showed anything.
Yeah. Right. Yeah, that's what we're thinking. But it's interesting that, you do have the one-to-one relationship and the very good selectivity against L5, but, I think to drive the SAR structure would definitely help.
Possibility.
Yeah, good observation. Okay, I think, is that it?
That's all for me.
Okay, thanks. Great update. We look forward to the structures, that's for sure. And it's exciting that they're starting to come in. So I think, Sue, you're up next. Do you want me to open the presentation, yours, or do you want to drive it from your end?
That's right. If you drive it from your end, please.
Okay.
Okay.
All right.
Thank you. So just start off with a general update. So we're now in a position to slightly increase the amount of resource. As you know, we've had some difficulties recently, but we've got some more available resource now. So we're able to complete some of the targets that we had in progress up until recently. So 2 of the original compounds that we were asked to make right at the start of the collaboration, we've now completed those synthesis, and the remaining 4 I would consider a probably lower priority based on how the SAR has progressed since then. We've completed the synthesis of the compound from the Merck patent, and 3 of the 4 compounds, well, 2 were from the Novartis patent and 2 were corresponding to methylpyridines from the Novartis patent. And the blueprint patent compounds have proved somewhat more challenging. They're somewhat more complex in the synthesis, but we've got several of those that are very near to completion, some in sort of purification. So hopefully we will have those fairly soon. So we will be shipping the 6 compounds that we definitely have completed in I think it's the last week of May that David had suggested, but we'll coordinate with the next shipment that's going from the OICR so that we can ensure that they'll get screened in the single batch. So we went back and looked at the ideas that we had that came out of the brainstorming, and looking at the work that was being carried out at the OICR. Obviously, we didn't want to duplicate any efforts. So we've been sort of reviewing where we can now fit in and make some new compounds. We're also going to start looking at some new ideas to come up with a second series, because clearly in the requirements of the project are to have a second series. We had some sort of preliminary ideas from our brainstorming, but they need to be worked up a little bit more. And I think now that we've got some of these competitor compounds prepared, we can send them to Alex for crystallization. I think that's going to help us to understand better how they bind, and that might enable us to come up with some ideas for a new series. So once we've got that information, then we can have some more brainstorming sessions and get a little bit more detail on ideas for a new series. So the other thing is that we have agreed to carry out SIP profiling of a batch of 5 compounds from the project. If you would like to make a selection of 5 compounds and send them to us, we can carry out a SIP profiling. And I think I outlined in an email to you, Jeff.
Yeah, no, that's exactly right. I think, we're ready to make a proposal at the end of this meeting, I think, for further profiling. So, yeah.
Okay, next slide, please.
You want to ask her? Sorry, Sue, there's a question?
How big is the panel? How big is the panel for second inhibition?
Sorry, I can't. Can you?
The question is, how big is the panel for the CYP inhibition?
Okay, so we can do, it's 7 isoforms that we routinely test. And it really depends on, we have to be a little bit flexible, because what we'll do is we'll fit it in with other work that's ongoing for various projects. We may either be able to provide you with a single dose, single concentration, or we might be able to do IC50s. It really depends on the work that's actually going on. So we can see how it goes from there.
Yeah, great.
Okay.
Yeah.
So next slide, please. Okay, so the original CRL targets that we had are shown on this slide. So there were 6 targets, 2 that on the left-hand side, well, one's completed and one's just about back from purification and will be ready to be shipped, may well be ready by the time we come to the next shipment. The remaining 4 targets, as you will see, 2 of them had target C has the 2-methylpyridine, which we've now moved away from. Obviously, one of the earlier ones did, but we've already made that before we had the new SAR data. But the target C I've indicated there, really, I'm not entirely sure that it would be a valid target to prepare now. On the other hand, you might be interested in the compound on the far right, which would be the 4-methyl analog of that.
Mm. Right. Yeah, no, I think that's probably a better one to make, yeah.
The other 3 targets, all based around this 2-piperazine, I can't remember the number of the compound, but it's the one that you were saying that we've now confirmed that it's not active. So I don't really think that there's any value in continuing with those targets, so I've put those on hold as well.
Yeah, no, we agree. I think that's a good decision. The 1212 wasn't active, so that ortho position, the ortho substituted pyrazine doesn't seem to be well tolerated. So we certainly agree with that. Yeah.
Okay. So next slide, please. So this is just a little bit more detail on where we're at with the competitor compounds. So in the top left-hand box, you've got the Merck compound, which was claimed to be 20 nanomolar on ALK2 and pretty selective over ALK5, but very similar activity on ALK1, which from the presentation earlier suggests that would be perfectly acceptable. That compound has been made and is ready to ship. The Novartis compounds, the two compounds with the amino group in them, where X, in the bottom left-hand box, where X is NH2, those have been synthesized, and one of the compounds where X is methyl as well. The other one is almost completed, so we won't stop that. As I mentioned earlier, the blueprint patented compounds, the synthesis has been a little bit more challenging, but we are nearing completion. We've certainly got the compound, the left-hand compound with the cyclopropylamide is in purification, and the compounds on the right-hand side are also nearing completion.
No, that's fantastic. Yeah, we look forward to seeing that.
Yeah. So some of those compounds will be in the shipment to RBC, but I think, Alex, how many compounds would you like to receive of this set?
So we had two ideas. I mean, the crystallography is obviously something we can do, and I think there we would probably just maybe go with the most potent ones in these series, unless you had standout ones you really want to try. We'll talk offline, but we're also interested in if we get the nanobret extended to the whole family. We're also talking about putting together some screening paper on the capabilities of nanobret, and this would be a good test set to kind of benchmark against the whole ALK1 to 6 family. So in that context, maybe screening all of them would be of interest.
Yeah, I think we should definitely screen them all, at least against the ALK2 nanobret and ALK5, right? I mean, just to benchmark them against our own compounds. So we should probably just send them all to Alex, Sue.
Yeah, absolutely fine.
Yeah.
Yeah, I'll sort that out with you offline then, Alex.
Okay. Do you want to ship them to Jeff, and then will Jeff forward them here, or you just ship them direct to us? Because obviously Jeff wants to send some to reaction biology as well.
Well, we'll be sending them direct to reaction biology, and to be fair, it's probably for us to send it direct to you, because we don't have to send it abroad and then for you to send it back again.
Perfect. Okay.
Yeah.
So we'll send them direct to you.
And are we having a meeting with you in June? Is that right?
That's correct.
Direct. So that would be another option. We could just pick it up then.
Yeah. Yeah, we can probably sort some of them out before that and get them to you sooner if you like.
Okay.
Let's liaise offline, shall we? Yeah.
Okay.
Okay. So yeah, if you move on to the next slide. So just looking at some of the plans. So I went back to consider what we could do, and I think the main focus, obviously, there's a lot of work has been going on at the OICR to look at substituted phenyl replacements for the trimethoxyphenyl. So I thought, looking back at a lot of the ideas that we had, maybe we could look at some of the heterocyclic replacements to just make a differential between what the two sites would be doing. Mm-hmm. So I thought that looking at, so we originally had compounds like M4K 1196, which had pretty reasonable activity. It's not as active as obviously 1062, but it still shows some interest. So can we replace that with some of the, rather than the indazole, have the benzimidazole, which from some of the modeling suggests that the groups will be in the desired, so the methoxy and the nitrogens of the heterocycle are in the preferred positions to interact both with the lysine and with gluten 248. Mm-hmm. So are you happy for us to work with these? Yeah.
Yeah, okay. No, these are great ideas. Yeah.
So part of the process will be sorting out what intermediates are commercially available and how easy they will be to synthesize, obviously, and that may limit the compounds, because if we've got to do a complex synthesis of the group that we're going to introduce, that's going to be more challenging. So we'll try and do the relatively straightforward ones first.
Okay, so that sounds like a good plan.
Move on to the next slide. So this was really looking at some other heterocyclic replacements, which also came out of the brainstorming ideas, where we might be able to replace that trimethoxyphenol with pyrazole, which can be in different orientations, and then would place the functionality in the position to interact with either the lysine or, as I said before, the glucophorate. So it's interesting that M4K 2017, which I think we talked about a little bit last time, is similar to the pyrazole compound on the right-hand side, where it's linked through nitrogen and then has an amide group. And we had docked that. I haven't actually got it on this slide. We've docked that, and it shows a reasonable interaction. So the picture is of the compound on the left-hand side with the hydroxyethyl pyrazole. That's correct, yeah. So that again gives an interaction with Glu-248. I think some of these types of compounds look to be relatively straightforward to make. So I think this again will give us something a little bit different from some of the targets that you've been looking at yourselves.
Yeah, no, that'd be great if some of these work, because you know, we are finding that trimethoxy more difficult to replace than anticipated. So.
Okay, so the next slide, please. So the other thing I was looking at was that there were a number of ideas that came out of our brainstorming session to replace the central pyridine. Now, obviously, we know that the 2-amino is the most active and the 2-methyl is a bit less active, but putting the methyl group in the 4 position gives really good activity. But also replacing that C-methyl by nitrogen in M4K 1210, I think it is, gives quite reasonable activity and good selectivity. So we thought about some other ideas. So on the left-hand side, in the sort of yellowy coloured box, we were also looking at using some information from the ICR series, where they have a quinolinone in there. So can you replace that by, in our series, by a pyrimidone? That puts carbonyl where the methyl is, and then would allow you to link through nitrogen rather than through carbon. Obviously, we don't know which way round it would be preferred to be. And on the right-hand side, so we'd be looking at introducing, so we know that if you just move the nitrogen one round, where you've got your cursor there, that's not active. But we have some chemistry that has been worked out in-house, where you can introduce, you can prepare these sorts of pyridazine targets. And also on the right-hand side of that box, you've got the pyridones, so where you've got the donor-acceptor pair in the different orientation from in the aminopyridine, and see whether those sorts of targets actually will give us, and we see that sort of pyridone in the Merck compound, of course, which we had earlier.
Good.
Okay, I think that's my summary. Any comments at all?
No, like I said, I think they're great ideas, and you know, hopefully some of those things pay off. Especially, you know, the trimethoxy is, like we say, is getting tougher to replace, it seems.
Okay, thank you.
Yeah. David.
Perfect, thank you. Okay, so I'll talk about some of the ongoing chemistry and here at OCR and some of our future plans as well. So just a quick recap, we started with the methyl up top at the 2 position, but we seem to lose 5 to 10-fold activity with that. And so we put in the 4 position, which gives better selectivity and retains the potency that we're seeing from the unsubstituted. The NH2 up top at the 2 position is good for potency, but we have very poor selectivity. So we've kind of moved away from both of those any kind of substitution at that position. We've been doing a lot of work trying to replace the trimethoxyaryl in the pocket side. Seems, as Jeff mentioned, a little more difficult than we initially thought. And we've been looking at a few different soluble lectin groups to try and improve potency and, more importantly, to improve selectivity there. I think there's a lot of avenues for that. So based on the work of Alexi and Greg, which they presented, I guess, 2 meetings ago, one of the methoxys seems critical. We don't see anything in the crystal structures, though. It just seems to be more of a space-filling interaction, van der Waals maybe. Removal of the paramethoxy was accepted, and in some cases beneficial. Maybe giving the other methoxy that would be in R1 a little more room to turn. And R3 seemed to be highly variable in their compounds when they tested against RIPK2 and ALK2 by RBC. So a lot of substitutions were tolerated, a lot of big groups. So in that vein, I made the 3,5-dimethoxy compound, and that showed really good potency against ALK2 and good selectivity. The selectivity, I don't know what that would be from, but I'll take it. But then on changing up and increasing the bulk, just making a series of ethers, we lose selectivity and we also really reduce our potency. So most of the compounds showed very, very weak potency, at least a 10-fold drop in each case. A couple inactive and a few, the 3 on the bottom right, are in the last batch sent to RBC. And we're sent to Alex as well, so we have the cell-based data on that.
Did we ask for a repeat on the 1071?
Yes.
Yeah, in this current batch?
Yeah.
Yeah, because that's the compound Alex highlighted as well.
Yeah.
Like, had that disconnect, right?
Yeah, we're getting it retested from the stock solution.
Yeah, perfect.
So again, we were looking at the Greg and Alexi's data where they have the amino up top, and a wider range of sulfonamides were tolerated with good ALK2 potencies, all around 10 nanomolar, with obviously a wide range of bulk associated. So we were just kind of talking back and forth about maybe the hinge, the angle of binding at the hinge is slightly different. And so that the directionality of our substitutions are changing a little bit. So R3 would be pointing towards, out more towards the solvent side, and where the wall might be a little firmer, and less towards the cavity further in, towards the lysine that's there. So based on that, we started looking at para substitutions, and these are some of the ones based on Alexi and Greg's compound that I had previously prepared. We're waiting on the RBC data, where there's no data built in, but it showed okay cell-based potencies, 200 to 300 nanomolar there. But a pretty steep drop-off when you're removing the amino. It could also potentially be a polar surface area interaction or dipole moment, dipolar effect, making the amide or sulfonamide there rather than a phenyl ether.
Could you compensate for that PSA increase by going back to the end method? When you make the sulfonamide, could you add the end method and might improve cellular profile?
Yeah,
That's a possibility. Yeah.
Nice. See what the RBC data looks like. Those positives, and then
And then the end to fix the properties.
Yeah.
So intrinsic potencies there.
Yes.
Yeah. So based on just what we've been talking about with the hinge binding angle maybe changing, as well as trying to get some groups pointing further into that cavity, we've been looking at changing the substitution on the ring, substitution pattern, whether it be 234 or 2345, different groups there, as well as substituting at R3 so that we're pushing in the para position out towards further into the cavity and maybe to try and pick up one interaction there. We're also trying to cyclize around and lock that head ring via some heteroatoms or maybe just a carbon linker back onto the core. So, yeah.
Have you considered putting just a pyridine in the middle position, the R2 position, or one position because of changes? So on the slide that you're showing right now, the structure is, where R2 is, have you considered putting pyridine there?
Yeah, I'm pretty sure I made the dimethoxypyridine headpiece, and it showed no biochemical activity, nor cell-based.
Oh.
Right, it's very, very sensitive in that area.
So some solvent side modifications we've been exploring. They all show pretty good potency and in some cases really, really good ALK5, ALK2 selectivity, specifically the one top right with the isopropyl. That really has great ALK2 potency, but very diminished ALK5 potency for selectivity around 300. As soon as you start changing it from the pair positions, though, we start to erode our ALK2 potency. So bottom left, 2042, we see a decrease. But as soon as you change to now a heteroaromatic 5-member ring with the pyrazole there, we have now better ALK2 potency. Although the ALK5 selectivity isn't all that great. We're still exploring what we can do here, different substitutions, and a lot of different things, trying to take some ideas from the many ideas out in the kinase space.
Do you know if that affects toxicity? Because there was a concern that methylation there was causing toxicity in vivo.
The methylation on the pyrazine you're talking about?
Yes. No, methyl.
Yeah.
Yes. Compounds like 2042, for example.
Yeah, I mean, so I think there was some toxicity observed in one case with an N-methyl, but in an IV arm where I think they were dosing 10 mg/kg in the IV arm, which might be a little high. I think, we'd rather see that dosed at 2 mg/kg and then, really get a feel for the toxicity that way. So, we're hoping that's not a general trend with the series, but rather than just an isolated incident with one molecule.
Yeah, I think it would be really nice to understand that, because this could be a place where, the alkyl group on your pyrazine could be a really good place to introduce some more additional groups to improve your solubility and your cell permeability and things like that. So understanding whether that really is a toxic effect of the N-alkyl pyrazine.
Right. You'll see. I mean, we're going to put some compounds in vivo, and one of them is this isopropyl analog. So hopefully we'll get a feel for that then.
The Novartis solvent side group also had that pyran ring that was coming off of their ferolidine, so I don't think it's necessarily a general effect.
I think that's an important point, because I think your CNS profile is going to be changed quite a bit between NH and N-methyl, so the quicker you sort that out.
Yeah, and it removes the hydrogen bond donor,
Yes.
right? Right away. So if we start adding them other places, like on the solvent side, then that'll allow us to do that. And we can hopefully tweak it seems like it's well tolerated for potency, so as Sue mentioned, we can really tweak properties by using that substitution. So,
Have you ever,
and it does seem to impact selectivity. So,
have you ever looked at toxicity in the cells? For example, in the cellular assay, just adding, 10 micromolar compound, whether it has any toxicity.
So we're just starting to think about getting data in proliferation assays. I think, Alex, maybe you've generated a little bit that you showed at the last assay or at the last meeting, but so we haven't put these compounds into anti-proliferative assays yet. But the cell-based assays that we have looked at, and Alex, maybe you can comment, but I mean, I think that the numbers we're getting are pretty reasonable for those compounds, and the curves look good as well. So probably not a lot of concern there, at least at this point.
Well, actually, we have some of our compounds recently just for the revision, and we didn't see any toxicity. But I'm curious about this methyl and methyl, what that does.
Yeah, no, I mean, it's something that we need to follow up on, that's for sure, before we move forward with those compounds.
Do we actually do a cytotox assay at all?
We haven't yet, but a cytotoxicity assay, an ACT or something. Yeah, I mean, we could certainly think about scheduling one.
I think it would be useful to know whether they are actually at higher concentrations killing the cells. I think it'd be useful information.
Yeah, I'll take note of that. TT assay. Okay. Yeah, I'll see where we can schedule that. Okay.
Okay. So beyond just some halogenation of the solvent side phenyl ring or insertion of a nitrogen to make it a pyrazine or a pyrazine there, we've also been trying to change some bond angles, introduce fused ring systems. The one on the right, or the one on the left at the top, you can easily do a wide range of productive animations there to try and tune some properties, increase solubility, as well as introducing methylene spacers, so benzylic, piperazine, morpholine groups there. And we're working on the tricyclic variant on bottom right, which is similar to the one where we're trying to tether back the head group. So a couple different ideas there that are all underway. So just to summarize, so we're still working to try and replace the trimethoxyaryl, which we think is going to be a liability. Don't know for sure, but we'd like something a little bit better. Try and pick up one extra cavity side interaction to replace the amino group that we lost. The sulfonamides and amides were being tried, just like Alexi and Greg showed. We don't see, we see okay cellular potency, not great. We'll have to wait for the RBC data to make any strong conclusions on that. And I'm shifting them over to the para position on that. The modifications to the core have proved pretty fruitful. We've tried the 4-methyl, which seems to be pretty good for the selectivity, and further modifications have been suggested or underway. And the solvent side groups seem pretty replaceable, so we have a lot of room to work there to tune properties, try and increase selectivity. A lot of kinases, changing that group can really give you a boost in terms of selectivity and potency. So
good. Thanks, David. All right, we're almost done here. Okay. All right. So I just wanted to remind everyone kind of what the key compounds are to date, and so some of the key observations that we've made. So M4K 1055, and I don't know, Michael, if you're still on the line, but this is the profile that we've generated for that 117 analog that you demonstrated a lot of very promising biology with in the FOP space. So in our hands, this compound is about 115 nanomolar against ALK2 at RBC, and it's very selective against ALK5 though. And in the cell-based assays, I think, there we see a much better correlation with the Boehringer data, and that it's, around 200, 250 nanomolar against, in the nanobread assay, but it's very selective against ALK5. Again, this, may not be potent enough and really the basis behind our project is to try to improve this molecule to drive that potency down while maintaining some level of selectivity that's close to this. So, as was previously mentioned, going from the 2-methyl to the 2-hydrogen here, we generate a compound that's now 18 nanomolar, still gives a good selectivity profile against ALK5, and that translates as well into cell-based selectivity. It's about 23-fold. So, the ALK5 potency is still around a micromolar in the luciferase assay. 2009, then moving the 2-methyl to the 4 position, gives us again a compound with about 13 nanomolar potency against ALK2. Not very selective against ALK1, but I think, I'm much more reassured that that's something that we can tolerate, and maybe we need to consider moving to ALK4 as the other ALK that we routinely screen, right? So that might be something that we change in the screening cascade. Good selectivity against ALK5 is 2009, and pretty good activity in the nanobread assay, and it's about 1.6 micromolar in the luciferase ALK5 assay for a selectivity of about 30-fold. And it's metabolically stable, and we actually move that compound in vivo, and at 25 mg per kg, we get around 1.7 nanomolar Cmax at about 6 hours. So, the compound looks reasonably good. And then we've gone on and tried to capitalize on that 4-methyl substitution by removing a nitrogen at this position relative to 2009, so 2043, shows similar levels of potency all across the board, perhaps a little bit better selectivity than 2009 in the cell-based assay. So we're up around 50-fold selectivity in that cell-based assay. But again, we should really firm these numbers up as we move forward. Introducing substitution at this nitrogen relative to 2009, so the isopropyl analog, again, it's about 7 nanomolar. In the RBC assay, it was very selective against ALK5, a little bit less so in the luciferase, in the cell-based assays. And 2044 certainly is a very potent compound, but again, it's not super selective in the cell-based assays, but again, very stable in the metabolic stability assays that we run here at OACR. This 2046 compound that introduces an ether linkage between the basic moiety and this phenyl group is well tolerated. It's of similar potency, but the metabolic stability drops quite a bit. So, I think that those are the most interesting compounds that we've generated so far in this project. And the next steps are to take kind of 1062, 2009, 243, 2044, and 2055. So those are the ones with the arrows on these slides, and I'll make these slides available so that everyone can go through the data again. But we propose that, one, we try to get X-ray structures for most, if not all of these. Some of them are pretty similar, but, we'll be more or less successful, with some of them, and probably not all of them. We propose that these are the ones that we move forward with the SIP profiling at Charles River, and thanks, Sue, for organizing that. We want to continue, making sure that we get hard numbers. So, consistently sending some of these for N equals 2, N equals 3, both at RBC as well as at the assays running at SGC Oxford. So, just to continue firming up the numbers so that we're really confident in the numbers that we're generating. We think, profiling these in the DIPG cell lines, both in terms of proliferation as well as in the phospho-SMAD. I think, this is something that, can be done both at the SGC as well as Jerome. I think, you're also willing to do some of this. So I think, these are certainly 5 compounds that we'd be interested in. And, we probably for those assays, we can give you a handful of others as well that we're interested in. I think 2022 is one that Alex took note of, that we should probably, think about that as well, just to make sure that it's on target effects that we're seeing. Yeah. And then, I think we're ready to move forward with at least some benchmarking pharmacokinetic studies. So the plan is to kind of move these 5 compounds into a full mouse PK study. We have quotes to be able to do this that are quite reasonable, and so we think we can move forward with this. And then we did get quotes to look at brain exposure for some of these compounds. So if we look at 2 time points after an oral dose, it's about $2,100 per compound. If we look at 3 time points, it's about $2,800 per compound. So it's expensive, but I think it'll be extremely valuable data. So what I'm proposing is that we get the full mouse PK of all 5 compounds first, and then use that data to select compounds, and then also to select the time points that we use in the brain exposure study. So I think I'd like to get this out as soon as possible, and to generate that data to be able to discuss at the next meeting, and to be able to set this experiment up. Either, I doubt we'll have the data for the next meeting, but probably the meeting after that. Of course, we want to continue the SAR, and I think both David and Sue showed you the direction that's going. I think everything looks promising there. And then we still need to figure out about the in vivo efficacy studies, where that's going to take place. I think that's critical, and we need to start looking at that more in depth. And I think there are certainly some labs out there running some of these experiments. I think Jerome has some interesting models that are under development. If some of those work out, then we'll be able to use that. And then, as Sue mentioned, we still need to consider a backup chemical series. But I'm really happy with the progress that's being made in the chemistry in the lead series, and I think demonstrating that proof of concept soon is still something that we want to do. This I thought was interesting, a piece of interesting news, but I don't want to take away from the last couple of minutes. I know Arish has a couple of comments to make on the website, but there was on April 19th, Dr. Nicola Vitanza out of Seattle's Children's Hospital was able to take patient samples that were treatment naive from a DIPG patient, was able to grow tumors in 20 mice. And so, I think this is really interesting, because most of the other models that have been generated to date have been used with treatment experienced samples, and so they've already been treated with radiation, and who knows exactly what that's doing to the cells, and what actually survives coming out of that. So I think this is an interesting advance, and perhaps we can try to make contact with Dr. Vitanza and see exactly what his research is all about. So with that, and I don't know, Arish, I think we need to, sorry.
I just have a couple quick questions. So what is the clinical setting? Is it treatment naive, or is it going to be heavily pretreated or co-treated with other drugs for this? Because that also impacts the CYP profile, target profile.
Right. So the only treatment out there now that's effective for this disease is radiation. So
they wouldn't be on other drugs?
They would not likely be on other drugs.
So therefore, CYP wouldn't necessarily be a concern?
Not necessarily, no.
Okay.
No, I think it's good to
it's more of an FYI.
It's more of an FYI.
Oh, okay.
Yeah. And, I mean, as the disease area progresses, it's very possible that other drugs will be given, right, by the time this gets to the clinic or to the market. So I think it's still good to try to minimize any potential. Yeah. Okay. Okay. So Arish wanted to give just a quick update about the website.
Yeah, sure. There.
Yeah. Do you have a presentation?
No. You said you can load the website on your.
Oh yeah. Okay. Yeah. Let me load it here. Yeah. Can everyone see the website?
Yep.
Yes, we can.
Perfect.
Okay, hi everyone. So I'll keep this short and sweet in the interest of time. So the M4K website underwent a major redesign recently to include more information on the company, the team, partners, and our mission. We also have a new logo you'll see here at the top left, which incorporates the open access, open science symbol, the open lock. It's a recognized symbol in the community for open science. M4K also has a new tagline or a mission statement, which is open science for children's health. I'll just go here. There's also a blog space on the website that will house all updates from M4K, any relevant news on open science and DIPG. Eventually, the website will also, and the blog will also direct visitors to M4K's shared open data repositories. Here. So M4K has several social media accounts. In the interest of time, I won't go into all of them, but I do want to point you to the M4K Pharma YouTube channel, which will have all of the recordings from our monthly meetings, as well as other videos. So here's an interview that Owen Roberts, CEO of M4K Pharma, did with Bio 2040, which is a blog site that looks into trends for drug discovery, and of course the lemon face challenge that M4K, SGC, and OICR did, to which Charles River responded epically. So that's great. And now we're waiting for the next video from Alex and Paul Brennan. Finally, so between the website and the social media outlets, I think M4K has a well-rounded online presence, so can help facilitate connecting with individuals and other organizations who might be interested in partnering with M4K and the team, or just interested in learning more.
Great. Thanks, Arish. So I think that brings us to the end of the meeting. Is there any additional questions, comments?
It would be nice, some of those benchmarks that Sue is preparing.
Yeah.
Those could kind of make it into some of the screening.
Oh yeah.
That would have to, some of the DIPG.
Oh yeah. No, we want to move those in as fast as possible, basically. So we'll Sue, yeah, we may have to talk to you about having some of those compounds shipped to us as well.
Yes, of course. Yeah, no problem.
Yeah. Okay, fantastic. Okay, so again, I think it's been a really good meeting. I think, again, we continue to make progress, and I think soon enough we'll hopefully be at the proof of concept in an animal model stage. So with that, I look forward to next meeting. Next meeting we will be in the UK, Owen and I. So.
And the rest of the team will be at UT. The
Right.
Toronto team will be at UT.
That's right, because OICR has scheduled its annual fire alarm drill during the next meeting. So everyone has to go to U of T for the next meeting. But it's gorgeous outside this time of year, so it'll be a nice walk for everyone. Okay? All right. Thanks, everyone.